Hi everyone, and welcome to our video on kidney failure. This is again part of our Module 5 series and the last one in the section on excretion. What we'll be looking at in this video are two sections of specification, 5.1.2e, which is the effects of kidney failure and its potential treatments, and 5.1.2f, how excretory products can be used in medical diagnosis. Okay. First thing, let's have a look at some of these diagnostic tests that we can actually utilise when there's something going a little bit awry with the body. Now, one of those go-to tests that you will probably experience at some point in your life when you go to the doctor is they're going to ask you to pee in a tube. And basically what they're going to do is then carry out a whole battery of tests in very simple ways on that urine sample. One of those tests will be testing for glucose, and the reason that they can test for glucose in your urine, because in theory, if everything is working as it should be, then all glucose should be reabsorbed by your proximal convoluted tubule, the PCT. Now, if obviously they're doing that little dipstick test on the urine and they're finding high levels of glucose there, then what that is suggesting is that your cells of your proximal convoluted tubule are not able to reabsorb all of that glucose. And that's sometimes an indicator of diabetes. Second test that they can do is testing for ketones. Now, ketones are again diagnostic tests used to identify if someone has diabetes. So they're just a family of chemicals that will occur in the urine if you are suffering from diabetes. They can also test for proteins in the urine sample. Now, if we think back to the video that we had on the kidney and how it actually works in that process of the ultrafiltration side, then what we said was with those three layers that we've got to have the substances passing through to get from the glomerulus into the Bowman's capsule, then any proteins with molecular weights over 69,000 should not be able to cross that basement membrane, and therefore they shouldn't be present in the urine. If we do have these proteins present in the urine, then there's a few things that might indicate. It's not a this is definitely what you've got, it's an indicator something has gone wrong, and then obviously further investigations would need to be done to identify exactly what. But presence of proteins in your urine might be that your blood pressure is too high, because if you've got too high a pressure in your blood, clearly that's going to be forcing substances through those holes with a greater pressure, so larger bits are going to get through. You could have an infection in your kidney, or we could have a problem with the filtration, so there could be damage to the basement membrane, for example. We can also test the urine sample for nitrate ions, and if there are nitrate ions present, typically that's going to tell us that there is a bacterial infection somewhere within the urinary tract. So as we can see, there's a whole range of different tests that can be carried out on that sample of urine. Now, they're not going to sit there and do an individual test for each and every one, because that would be really quite time consuming. And if you've actually had a urine test at the doctor's, then you know that they don't do that. They literally get one little dipstick out, so it's like a little plastic strip with these little foam pads stuck on it, and that's what they're then going to put into the urine sample. And what these usually have are about 10 different factors that can be tested for at once. So just by dipping that little plastic strip in there, those 10 different little pads all have different reagents, and then all they're going to do is compare those colours to what they would expect on the little colour chart here and identify what you've got. So you'll have different colours and different rows for whether there's nitrates, proteins, etc. And then that's going to give us a good idea about what might be going wrong. One of the other things that we can test for using urine, and something that you can actually do a home test for these days, is pregnancy testing. Now, one of the things that hopefully we have looked at back at GCSE are the basics about pregnancy itself. What actually happens is once you've actually got that fertilized egg, if it then implants into the wall of the uterus, then it's going to start to obviously undergo that process of mitosis to produce more and more cells. Now, what we're going to find is that little embryo, as it's developing, is going to produce this stuff here, HCG, human chorionic gonadotrophin. And basically, what these pregnancy test kits do is they're testing for HCG in the urine. Because if that embryo is implanted, it's going to be producing that HCG, and that's going to end up in the urine sample of that now expectant mother. And the way that this works is by using these things called monoclonal antibodies. And in theory, you had a look at those at GCSE if you did higher tier papers. Now, monoclonal antibodies are basically antibodies that we've created that are complementary to something we have of interest. In this case, HCG. So the way these little pregnancy tests actually works then is we have got our little antibody stuck onto a little blue bead, okay? And then a bit further along our strip, we've got this little window where we've got the antibody molecules stuck down in that little strip. So they're kind of stuck onto the backing, if you like. So what happens is when you actually pee on the end of the stick, and you'll be peeing on this little end down here, then if HCG is present, it's going to bind to the antibodies that are stuck onto those little blue beads. And because we've now got a liquid on a foam pad, what happens is it then starts to soak up the actual bit of foam. So what we see is we get movement. And those little blue beads are going to be carried along with the liquid up that little test strip. When we get to this second window here, where we actually have our antibodies stuck down in a little line typically, then if we've got our little antibody bound to those blue beads, they're also complementary to the antibody that's stuck down. So they then bind there too. So all that happens is we get this little line of blue beads all being stuck down so that when you eventually look at it, you get a blue line on that pregnancy test. What we tend to see is that these days the pregnancy tests actually have two little windows. And what we have is our first one is the test line, and the second is going to be a control line. And the whole purpose of the control line is to let you know that the test actually did work. Now, obviously what we're going to see is these little blue beads are going to move up the actual bit of foam, and only if they've got the HCG would they bind on the test line. What we need is to then have them binding onto this control line regardless of if they've got that hormone stuck to them or not. It's just to let us know that the urine sample actually did make its way across that little foam pad, and then we'll form a line in our little control section. It just lets us know that the test is valid. In terms of the potential outcomes of these tests, so we've obviously got our little test here, and if we get a line in both the test and the control, that's going to be a positive pregnancy test. If, however, we get a line in the control but not in the test, that's a negative test. And if we then get one where we've got a line in the test but the control has nothing, then that is invalid, because if we don't have a line in the control, something has gone wrong. Just like if we've got this with literally no lines on it, also an invalid test. So in either of these cases where the control line hasn't appeared, we can't count that as a valid result. It's only if we've got the control line that we know that the test has worked accordingly, and therefore we can look at the test line to determine is it positive or is it negative. So if these kind of questions come up on multiple choices, usually there's a whole load of other bits in there, but narrow down your answers first of all. Look at it, identify whether it's positive or negative, and that's going to probably eliminate half the answers on that multiple choice question for you. Another use of urine testing is tending to be associated with sporting events. And what we're looking for here is testing for different anabolic steroids, which athletes will use to try to give them an edge in these competitions. So you'll hear about this every time we have Olympics. There's always going to be someone that fails the doping test. And all we're doing is taking these urine samples from those athletes, and then we're going to be testing the actual urine using either gas chromatography or mass spectrometry. And what we need to make sure of is that we're taking these samples close to the actual time of the event. So typically what you'll see is whoever gets first, second, third, etc., they're then going to be taken off to provide a urine sample, and that means it's going to be very close to the time of the event that we get that sample, because the drugs themselves actually break down quite quickly. They've only got a half-life of around 16 hours. So if you would say, oh, it's all right, we'll take a sample a day or so later, the drugs will already have started breaking down and therefore may not be detected. So the second bit of spec we're looking at today is all about kidney failure. And when we're thinking about the causes of kidney failure, there's not just one. They can fail for a variety of reasons. And the quite serious thing about them is that if your kidneys fail, if we do not provide you with treatment, then death is going to follow quite quickly, because what we're then not able to do in our body is regulate those different electrolytes, so the sodium ions, the chloride ions. We can't regulate the amount of water within our blood, so the water potential of our bloodstream is going to be completely out of whack. And as a result of that, combined with the fact that we can't remove some of these waste products like urea effectively, basically we're just going to be poisoning ourselves with the waste products, and everything is then going to be out of balance, causing other issues elsewhere in the body. So the way that the doctor will actually work out how effective your kidneys are functioning is by using something we refer to as the glomerular filtration rate, or the GFR. Now, as that name suggests, glomerular referring to the glomerulus, and the filtration rate is basically how quickly this region is filtering your blood, hence GFR. So what we're normally looking for here is between 90 and 120 centimeters cubed per minute. If you've got chronic kidney disease, then what we're looking at here is a value that's going to fall below 60 centimeters cubed per minute. And if you're in kidney failure, so this is the extremely serious moment, that's where your GFR is now below 15 centimeters cubed per minute. So you can see that's actually going to be 6 times lower than the lowest level for normal. Now, you don't have to memorize those numbers. If they want you to use them, they will give them to you in a question. But let's have a little moment just to remind ourselves about the correct notation when we are writing, obviously, our units here, because writing units is something you are probably going to have to do on one of your A-level biology papers. Do notice how in each case, what we've got is the time with the sort of superscript -1 there. And remember, when we're talking minutes, don't put M. M stands for meters, okay? So you've got to have min for minutes. Whenever we're doing our time for a rate, then it's always got to be whatever the value is for the unit, and that always goes to the -1, okay? Please don't write it as other things. Don't put your little slashes in and things like that, because that's not always accepted on the exams. So you've had your GFR rate measured and the doctor has turned around and said, okay, you are in early stage kidney failure. What do we do? Well, there's two options really. You can have dialysis or you can have a transplant. If we tackle dialysis first of all, because typically this is what people are going to be on first of all anyway, even if they're going to end up having a transplant later on, it's highly unlikely that they're going to be able to get a kidney there and then. So dialysis is almost certainly going to be where people end up first of all. And dialysis is where we're going to separate large and small molecules using a partially permeable membrane. And we've got two types of renal dialysis that we can use: hemodialysis or peritoneal dialysis. Let's have a look at hemodialysis first of all then. If we start by just looking at the name hemodialysis, hemo relating to hemoglobin. So basically this is dialysis using the blood from your body. We've got a little diagram on the right hand side here that's going to show us how this works. So the first thing is we're going to obviously have some kind of lovely plastic tubing inserted into one of our arteries which is then going to allow us to remove the blood from the body and pass it into the machine. So we can see we've got our little entry point down here. So they're going to insert a needle, and that's then going to connect into this little plastic tube that takes your blood from the artery, and we monitor the pressure there because we need to know what pressure this blood is coming out at. It's then going to pass through this little pump, and then it goes in to this dialyzer. But before it gets in there, we're going to add this stuff called heparin, and you can see it in this little syringe here. Now, heparin is just a chemical that we're going to use in order to reduce the risk of your blood from clotting. Because when blood is out of the body, if you cut yourself, then it's going to start to clot. So we don't want blood clots because blood clots are bad for your health. Therefore, we add heparin to this, and that reduces the risk of blood clots forming during the process of dialysis. So once the heparin's been added, it goes into the dialyzer here. And basically what we've got in that dialyzer are a series of tubes of dialysis membrane that it's going to flow through. Now, surrounding those little tubes, we've got a fluid called dialysis fluid. And the idea here is that we've got the blood going one way and the dialysis fluid going in the opposite direction. So we've set up something we've heard about in other areas of biology, a countercurrent. The whole idea behind a countercurrent is to maintain steep concentration gradients. So as this is flowing through those dialysis membranes, what's going to happen is those substances that we need to remove are going to be able to pass through the membrane. The bits we need to keep, like the blood cells, etc., remain inside because they're too big to pass through. And then the blood continues to pass through some tubing. It goes through an air trap and air detector because we, again, need to avoid killing our patients. And having air bubbles going into the bloodstream is very bad for the health. So we've got a little air trap and air detector that monitors and removes any air that's present, and then the blood re-enters the body. Let's have a little bit more detailed look at what's actually happening inside that dialyzer. So we've got a zoomed in little diagram of it just here, and what we have then are those little lines in the middle are going to be our dialysis tubing. We've also got that dialysis fluid, remember, that surrounds those. And the key thing about that dialysis fluid is that it's got all those substances that the blood requires, like glucose and sodium ions, in the correct concentrations. So obviously we've done many studies, we know what the concentrations of those things should be. And therefore, by creating a solution that matches that concentration means that they're not going to be pulled out of the blood unnecessarily. It also means that if there's a lack of glucose or a lack of those sodium ions in the blood, then they will just diffuse across that membrane into the blood. Things like urea are not present in the dialysis fluid, and therefore the concentration gradient is moving out of the blood into the dialysis fluid, so urea diffuses into the dialysis fluid. If we have a little look, we can see that the blood goes in at the top and flows down to leave at the bottom, whereas our dialysis fluid goes in at the bottom and comes out near the top. So that's our countercurrent system, all about maintaining those steep concentration gradients for those substances that will need to be crossing that membrane. Okay, option 2 of our dialysis is what we call peritoneal dialysis, or PD. Now, peritoneal dialysis is where we're going to utilise a catheter, and we're then going to use that catheter to be able to put dialysis fluid into the patient's abdominal cavity. So the idea here is we're kind of using their abdomen like a dialysis machine, I suppose. So what's going to happen is if we fill that abdominal cavity with dialysis fluid, then substances are going to diffuse across the peritoneum, which is the lining of the abdomen, and then that's going to allow us to balance obviously all those key bits that we need to ensure are in the right concentrations in our blood. We can then just drain the fluid from the abdomen itself. If we think about one of the key differences between peritoneal dialysis and hemodialysis, then the peritoneal dialysis is going to remove less waste than the hemodialysis. If you just think about the setup of it, then with the PD, what we're doing is we're just putting fluid into the abdominal cavity. There's none of this countercurrent system being set up. It's not being refreshed. We've literally just put the fluid in and it's going to stay in there for a while and then we remove it. So obviously that means that as substances start to enter that dialysis fluid that's in the abdomen, that's then going to obviously reduce the concentration gradient. So it is a bit less effective in some ways than the hemodialysis. If you are undergoing dialysis, then it's not just a case of you can just have your dialysis treatment and just carry on your life as normal. You will have to make other modifications, and one of the key things is that you've got to look at your diet. And in reality, you are going to have this very carefully controlled diet, which is going to very much be focused on minimizing the amount of urea we produce. And if we think back to the earlier work in module 5, we talked about urea being produced from excess amino acids, and therefore that comes from proteins. So we've got to monitor that protein intake. Second thing is you've got to reduce the salts going into your body. So typical salt is made of just sodium and chloride ions, and therefore if our kidneys are not able to function properly, we're not going to be able to balance our sodium and chloride ions properly. So we need to reduce that salt intake in order to reduce the strain that we're putting on this system that's not working as it should do. If we now have a look at a quick comparison between those two types of dialysis available to us. Hemodialysis, first of all, each treatment is going to take between 3 and 5 hours, and you're going to need that 2 to 3 times a week. And the key thing about this hemodialysis is it requires specialist machinery. And so in order to have that treatment, you are going to have to go to a clinic, and you will have to remain in the clinic for those 3 to 5 hours of your treatment time, 2 to 3 times a week. If we then compare that to the peritoneal dialysis, this one only takes 30 to 40 minutes per treatment, but you are going to have that treatment 4 to 6 times per day. So obviously, because this one, as we said, is less efficient at removing those salts and the urea, etc., then you do need that more regular exposure to that dialysis fluid. There is a big positive to it though, in the fact that once they've actually inserted the catheter, then you can just take control of that addition and draining of the dialysis fluid. And you can do that wherever you like. So to a certain extent, peritoneal dialysis allows you to still live a relatively normal life. So you could just fit this around your typical work schedule, because you could just do that while you're in work. Not like the hemodialysis, where clearly you've got to travel to wherever that clinic is and remain there for the duration of your treatments. So peritoneal dialysis does have some advantages in terms of giving you a bit more freedom and flexibility in your everyday life versus the hemodialysis, where clearly you've got to be near that clinic all the time. The other option, if we're avoiding dialysis, is a kidney transplant. And to a certain extent, the best option is a kidney transplant, because it removes all of that need for these regular treatments, and people just feel much better if they've just got a functioning kidney as opposed to having to utilise dialysis of whatever form. There are some disadvantages though. Obviously, if we're talking a transplant, then you are going to have to take those immunosuppressant drugs, because otherwise your own immune system might start to attack that transplanted kidney and therefore destroy it. If you're on immunosuppressant drugs, that does mean you are at greater risk of other opportunistic infections, remember. We're not talking about a quick 5-minute in and out operation here. This is major surgery. They are going to be removing the defective kidneys to replace with functional ones. We will also need to have that regular follow-up, just in case there are any signs of rejection, so that obviously we can take steps to resolve that, so your kidneys don't just shut down unexpectedly. And I suppose one of the biggest ones with any of these transplants are just the issues with finding a match. We can't just take any old kidney we happen to have lying around and just insert it into any old patient. we've got to make sure that we've done that tissue typing to make sure that there's minimal risk of rejection. And the problem is that we don't have enough donors. Even though obviously you can donate a kidney as a living donor, not everyone is willing to do that, because even if you've got 2 fully functioning kidneys and you're willing to donate 1, that does then put you at greater risk yourself later in life, for the simple fact that you've only got 1 kidney yourself. There's no backup plan for you at that point. So those people that do those living donations are truly amazing human beings, and I've got a lot of respect for them, but that's not for everyone at the end of the day. So we've got some downsides to those transplants, but obviously if we can do it, it is the best option. And one of the things that's obviously happened in the UK to try to rectify these issues with finding matches and the lack of organs that we've got is that people are automatically enrolled on that donor register now. You have to opt out rather than opting in. So increases the number of donors and the number of organs available for those that truly need them. I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next video is uploaded, and head on over to the website where you can find an array of other useful bits of information to help you in your A-level biology studies.